Well, good afternoon, everyone. It's 12 noon. We're going to get started. Welcome to today's webinar, Maritime Mosquitoes Monitoring, Tracking Range Expansion and Mosquito Populations in Nova Scotia. This webinar is brought to you by the Canadian Public Health Association as part of our Infectious Disease and Climate Change project. I'm Greg Penny, the Director of Programs with the Canadian Public Health Association, and I will be the moderator for today's webinar. I want to begin by acknowledging that the Canadian Public Health Association and myself are located on the ancestral and unceded territory of the Algonquin and Anishinaabe people. They have been guardians of this land for millennia, and we are grateful for the example their stewardship provides. CPHA is committed to working with all First Nations, Inuit, Métis peoples and their governments in realizing meaningful truth and reconciliation. So I want to start by actually just taking a quick minute to let you know about the poster contest that CPHA held. These are the winning posters for the contest. The contest was for grade 6 students and gave an opportunity for teachers and students to learn about climate change and the impact it's having on the spread of infectious diseases like Lyme disease and West Nile virus in our communities. Students across Canada could submit a hand-drawn poster to bring attention to climate-sensitive infectious disease for a chance to win a Chromebook and other great prizes. You can see all of the winning posters at cpha.ca/contest. So we are recording and we will be sharing the slides after with everyone once we post it. And with that, I'll actually introduce you to our speakers. So we have Dr. Laura Ferguson, whose research is centered around understanding how changing environments alter the outcome of host microbe interactions with a focus on arthropod vectors such as mosquitoes and ticks. Dr. Ferguson completed her MSc at Acadia University, studying the effects of blood parasites on mosquito physiology and behavior, her PhD at Western University, studying the impact of winter and low temperatures on insect immune systems and infection, and a Killam postdoctoral fellowship at Dalhousie University, studying the overwintering biology of black-legged ticks infected with a causative agent of Lyme disease. Currently, she is an adjunct professor at Acadia University in Wolfville, Nova Scotia, and the lead of a mosquito surveillance project funded by the Public Health Agency of Canada. And Laura is joined by Taylor Swanberg, who is currently a master's student at Acadia University. Taylor's research focuses on detecting the spread of invasive mosquitoes and determining the disease potential of mosquito-borne pathogens in Nova Scotia. She is particularly interested in investigating if molecular techniques of species identification from water samples, environmental DNA, can be used as an early detection method for invasive mosquito species within the province. Taylor completed her undergraduate degree at Dalhousie University in biology in 2016 and has since then worked on both entomology and genetic research. Taylor especially enjoys fieldwork, traveling abroad, and volunteering for conservation projects. And with that, I will turn it over to Laura and Taylor.
Wonderful. Thanks so much, Greg. I'm just going to share my screen. Classic Zoom intro. Where's the share button? Okay, here we go. Okay, so thank you again, Greg, for that lovely introduction, and thank you to everyone who's joining us here today. So Taylor and I are coming to you from Mi'kma'ki, the unceded territory of the Mi'kmaq. And today we're going to tell you about the surveillance project that we've been conducting throughout Nova Scotia. And I've called it the Maritime Mosquito Monitoring to include the maritime provinces in general, although we're focusing on Nova Scotia for the moment. A lot of what we talk about is very applicable across the maritime provinces. Some of it's applicable across Canada. But really we have sort of a focus on these provinces, and we share a lot of mosquito species between them. So we're going to talk about all of this today and move through. First, Greg did a lovely introduction. I thought I'd just sort of say very briefly again, I'm I'm Laura Ferguson, and I do focus on host microbe interactions, particularly in mosquitoes and ticks. And I have a penchant for understanding their overwintering biology as well, and you can tell that really excites me and very few other people often that have to do field work in the winter and like my son here, not enthused. And then Taylor, of course, just a wonderful graduate student who's leading the fieldwork portion of this project, and just a passion for conservation biology and entomology and molecular biology. So we've been having a lot of fun working together. And of course, we're joined by a really stellar group of collaborators on this project as well that I just wanted to take a second to mention. So we have Dr. Kirk Hillier, Dr. Russell Easy, and Dr. Todd Smith as well, who are all contributing to this surveillance project. So I first wanted to start off by just taking a minute to talk, or a few slides, I just talk about mosquito biology in general, because there's such a lot of variation in mosquito biology across the globe and the impact that mosquitoes have on our lives. And so it's important to take note of what leads to this variation, because this is really important for how we understand how mosquitoes are going to move across the globe and how our disease landscape is shifting with climate change, for instance. So we all know mosquitoes, we've all had them buzz in our ears and put calamine lotion on the welts, and you know, they're pesky. But we do have over 3,500 species of mosquitoes across the globe, about 80 in Canada, and as you'll see, about 30-ish within the Maritime provinces. And so we've been relatively protected here in Canada by infectious diseases that are spread by mosquitoes historically. But they are a huge problem across the globe, of course. Mosquitoes contribute to over 1 million deaths per year, and they vector a number of viruses and parasites that are quite devastating to human life, of course. So many different things are going to contribute to where these mosquitoes are living, what kinds of pathogens they're carrying, and the impact they have on their lives by the diseases that they carry. And so I'm going to talk a little bit about, very generally here, what we're concerned with now as climate change is progressing, is this idea that the mosquitoes are going to be experiencing shifts in temperature temperature and precipitation, among a variety of other things that we'll talk about as we go through, that are going to impact where they live in the world, where they can live. And some mosquito species are expanding their range across the world. And so one of them, if you're familiar with this area of research, of course, you will have heard of Aedes aegypti being one of these species that's really rapidly expanding its range across the globe. And we're looking at, in this map here, kind of a worst case climate change scenario, unmitigated climate change. Really important to take note of this warning, where we actually see that this map is showing us the number of months per year when this mosquito could inhabit these areas and transmit really devastating viral diseases. And you can see as you move up that even Nova Scotia, New Brunswick, and up through Canada, we're seeing even potential for this vector to establish and transmit disease. So this is causing concern, of course, because this means, again, that this disease landscape is shifting. So to understand this, we need to be able to track mosquitoes. And to do this, we need to understand a bit about their biology first to start off with. So mosquitoes have this complex life cycle where they spend half their time in the water as juveniles, and then the rest of their life out in the terrestrial environment as these blood-feeding female adults, at least, that we're most familiar with. So many species lay their eggs sometimes directly on the water, or they're going to rely on rainwater to wash their eggs in. But nevertheless, every species is going to require some kind of aquatic conditions. Those eggs will hatch into larvae that grow through various stages, and then eventually become what we call pupae. They're kind of these little commas that bob up and down in the water there, before then eventually emerging as these adults, again, that we're a little more familiar with. Now, once they emerge as adults, these are the stages that we're concerned about in terms of spreading disease to humans. And you can enjoy Taylor's wonderful drawing of a mosquito and my very rudimentary PowerPoint drawing of a blood vessel running through an animal. So this is the stage, of course, where these mosquitoes need to access blood. And it's just the females that are feeding on blood. They require this blood because it contains a lot of really nice protein that's going to help them make their eggs. And so go on and make all of their babies. So this is important to note that it's only the females feeding, and that, as I'm going to talk about, these insects are going to feed on different species of things as well. Now, also important to note, again, the males, they're just feeding on sugar. They actually have some beneficial pollination roles. Some of their species of orchids that are only pollinated by male mosquitoes. So they do have some roles in our ecosystems that we might not think about otherwise. And females are also feeding on, I might have said pollen there for a second, I meant nectar. And then we also have females feeding on nectar as well. They require that sugar. It does help them to produce more eggs, and it gives them a lot of energy to fly as well. Flight is really demanding when you're trying to fly around and find a host to blood feed on. So also important aspects of their ecology. Now, of course, we're familiar with mosquitoes feeding on us. So we are important hosts, especially when it comes to public health concerns, because we are concerned about what kinds of parasites and pathogens that mosquitoes can transmit to humans and make us sick. Mosquitoes feed on a very wide range of animals, and this is going to be species dependent as well. So some species of mosquitoes are going to be really specialized and pretty much only feed on humans. We're going to have other species that are a little bit more generalist, and they're going to jump from one animal to another, because these mosquitoes are going to be able to take multiple blood meals during their life, which is really important when we start thinking about, in particular, things like West Nile virus or equine encephalitis virus, that's going to jump between different animals in order to get to a human. We need it in a bird, for instance, to then jump to a human through this multiple blood meal scenario, or through establishing in a host and then having new mosquitoes transmit it on. So important to note what these mosquitoes are feeding on, where they're specializing, and whether we expect to see jumps in pathogens between these different hosts. Oops, jumped ahead there. The other really important thing to consider here, again, this helps inform us about where we expect to find mosquitoes. It helps us understand where we need to perform surveillance, is the idea that they're going to inhabit different areas depending on what species we're talking about. Like I said, we've got about 30 plus species here in the Maritimes, and they all do things slightly differently. So when we want to set up surveillance, or we want to understand mosquito biology, we have to know a little bit about where we're going to find them. And so, like I said, they require water for their young to live in, for these larvae to live in. And depending on which species they are, you're going to find them in different kinds of water. Generally standing water, they like things to be still, and they don't want to live in a spot where there are a lot of things like small fish to gobble them up, or dragonfly larvae that might completely decimate their populations. So you're going to find lots of different species in ponds and especially wetlands, and sometimes salt marshes, roadside ditches. And then, of course, we have container breeding species as well. So we have species that we often find in tires. And oh, I forgot to ask Taylor for the wonderful picture to put this up. My most favorite aspect of her fieldwork right now is that there's this lovely area in Nova Scotia where there are all these old toilets piled up. And this seems to be a very wonderful spot to collect mosquitoes and attract a lot of attention about the project in general. But in any case, these containers become really important. First of all, tires tend to be one of the ways that mosquitoes actually invade into new areas, which we'll talk about in a minute, but transportation in tires in shipping routes is really important. And we do find that they continue to use tires as these nice little locations to lay their eggs when water pools inside of these unused tires. And then we also see, for instance, containers in people's yards. So down in the bottom right-hand corner there, you can see these are actually containers out of my own father's yard. He's got this great yard full of mosquito habitat that I go and exploit and then try and dump out all the containers afterwards. But in any case, they can take advantage, certain species anyway, can take advantage of even very shallow water. So you can see a bucket there with just a very thin film of water on top, and certain species can reproduce quite quickly. And so even little areas like that are going to become really important to understand which species are there and whether those are species that transmit disease. And a little bit of spoiler alert as we'll get to, that certainly they tend to be the species that we are worried about the most. Now, mosquitoes are ectotherms as well, and this is important for their distribution in the world and how we think about where they're going to go and what's going to change with their vector capacity as we move through climate change as well. So what I mean by ectotherms is that their body temperatures essentially are going to be the same as whatever is happening in the environment as well. So when it's cold outside, the mosquito is cold. And when it's hot outside, the mosquito is hot. And this has direct impacts on things like how fast the enzymes in their bodies work and how well their cells function and things like that. And so direct impacts then on how quickly they can grow, whether or not they can vector a particular disease, and of course impacts on the pathogens themselves that we won't really get into today. But in any case, just this idea that temperature is extremely important for mosquito distributions. And so of course, with climate change and changing temperatures, this has a huge impact on where we're going to find them. Typically, we think of with ectotherms, including mosquitoes, that their northward distributions are limited by low temperatures. So winter tends to be what sort of keeps a lot of different species at bay, and probably a lot of what has protected us in the past against certain or a wider variety of things like arboviruses and other things that have been transmitted by mosquitoes or even ticks in the past. But we're starting to see that shift. So you can see on the map on the left, these are our mean winter temperatures across North America here. And so you can see even up into sort of Nova Scotia and through those maritime provinces there, you can see that we're moving towards some milder winter temperatures. And so this is going to invite mosquitoes that were typically limited in their northward distribution by our low temperatures to move in a little bit further. And so we're looking at the potential for more exotic species to move into these areas. And then the other important thing to contribute or to consider as well is precipitation, because like I said, mosquitoes are going to require those high levels of water in the environment to lay their eggs and have their young develop. So you can see on the map here as well, looking at precipitation across the board in North America. And you can see, again, sort of in these maritime provinces off towards the right here, that typically we're seeing a lot of, we're typically a precipitation heavy area, nice and humid, nice and warm. So good mosquito habitat. And again, you can sort of see this in this map. If we zoom in on these provinces, we're considered a warm summer, humid continental area of the country. And this has definitely implications for how welcome we are for mosquitoes who require a bit of warmth to grow, nice little water bodies, and lots of humidity to be able to survive and transmit disease. So now that we're zooming in a little bit, let's talk a little bit too about where mosquitoes could come into these areas and why we're concerned. So the other thing about this area of the country is that we have a few different ports of entry for global entry of mosquitoes into this area. So if you see the black arrows, so first, we have land borders with the United States into New Brunswick. And so definitely there's potential for mosquitoes to move through land transportation from more southern states or more southeastern states up through New Brunswick. And then of course could potentially then pass into Nova Scotia as well, which is potentially one way that we think some species might be hopping across. And then we have ports of entry as well, marine ports of entry from international shipping routes as well throughout Nova Scotia. And we definitely see that these can be huge impacts for how mosquitoes enter into new areas as they're shipped around the globe. Again, in things like the international shipment of tires, which seems to be one of the main ports of entry for things like Aedes japonicus, which we'll talk about in a minute. So the maritime provinces really are situated in this area where we're a nice spot for mosquitoes. And we have a few different ports of entry that could invite new species to move in. And recently we are tracking a little bit more and showing that Nova Scotia is indeed warming as well. If we zoom in a little bit more now, just looking at Nova Scotia, but really we are starting to see that our average temperatures are increasing overall, and that we're seeing that we're having longer frost-free periods of time throughout the year as well. So again, creating more conditions for mosquitoes, longer growing seasons, inviting them in. And the rate of warming across Nova Scotia is very site specific as well. So you can see on this map across Nova Scotia, essentially they're just looking at different regions where we're seeing differences in the average rate of warming over the last 60 years. And so this is important because we may then end up seeing pockets of Nova Scotia that become a little more friendly to new species of mosquitoes before others do, if we're sort of straddling thresholds for these mosquitoes to be able to survive in our climate here. We also have a lot of different ecozones across Nova Scotia as well. And Taylor's going to speak to this a little bit more in just a minute. But I just wanted to draw your attention to this idea too, that again, we have very region-specific areas. Even though we're a small province, we have very region-specific areas that have differences in precipitation, differences in mean temperatures, differences in elevation, and of course, differences in population as well. So this map is showing population densities, and this is even back in 2016, but you can see wherever it's sort of red on the map, sort of this darker red color, these are indications of areas that are more heavily inhabited within Nova Scotia. And so you can see sort of areas around the HRM. I'm just really realizing I don't have a pointer to point, but I'm so used to Teams instead of Zoom. But in any case, there are some areas there where you can see sort of around sort of the Halifax Regional Municipality, through the Annapolis Valley, up through Truro, Antigonish, Sydney, etc. So we're seeing these areas that are more heavily human inhabited as well. And so this is important because we need to know whether our disease-carrying mosquitoes that tend to feed on humans are concentrated in these areas of high population. So what is the disease risk in those areas? And then the other thing, which I'll speak to towards the end of today's talk as well, is what may be the impacts of a lot of development in these areas as well. So the HRM, for instance, in Nova Scotia is one of the fastest expanding city centers in Canada. There's a lot of development happening here. And so this has impacts, this human disturbance on the natural world also has impacts for things like mosquito populations and whether we're selecting for more human biters and disease-carrying mosquitoes as well. Okay. Oops. Seem to be frozen. One second. There we go. All right. Okay. So this, of course, leads us to our need for surveillance in Nova Scotia. I've talked a lot about now, but which is why we're sort of a nice, perfect little spot for mosquitoes to come and establish. But surveys in Nova Scotia are actually relatively rare. So back in 1979, we had one in 2001, 2002, and then now. And so we're going in about 20-year bursts of surveillance. And this is creating large gaps in what we know about which mosquito species are here. And mosquito species can, they often reproduce several times a year and could establish and spread through a small province like Nova Scotia in particular quite rapidly. And so we're really doing ourselves a disservice by not having more regular monitoring programs. But as Taylor well knows, and maybe we'll speak to, is that this sampling is really intensive, trying to go out and make sure that we're capturing all the different species of mosquitoes that live in Nova Scotia. And so it may be this intensity of sampling that prohibits sort of more regular sampling opportunities throughout the province. So we're also interested then in whether or not we need to do things like update our techniques. And this is, again, really important because we're already seeing some of the impacts over the last couple of decades of our changing disease landscape associated with mosquitoes. And not even necessarily new mosquitoes moving in, but even things like West Nile virus and equine encephalitis virus really only showed up within the last 1 to 2 decades in this area. And so these are relatively new, they are transmitted by mosquitoes, and we have to be able to keep track of them and understand what's going to shift as we enter into more pronounced climate change. So our main goals with this project then are to catalog all of the different mosquitoes that we have in Nova Scotia right now. Are we seeing big shifts over the last couple of decades? What does that mean for arbovirus transmission throughout Nova Scotia and into the rest of the Maritimes as well? And then the other aspect of this that Taylor's going to speak to is that we're also looking to pilot eDNA and community participation in surveillance programs as well. So this idea that insects are leaving behind bits of their DNA in the environment, and mosquitoes, this would be in their aquatic habitats as they transform from the larvae and pupae into adults. And so we have the potential here, this has been successful in other areas, to go around and sample water bodies and perhaps have community members just sample water bodies all across the province in their areas, and then sequence the DNA in these samples to be able to understand which mosquito species are there. And if we can do this in a very sort of cost and time effective way, perhaps then we can implement more regular sampling in this area of the country. Okay, so based on that background, I'm going to let Taylor talk to you a little bit about the results that she has so far from her first year of sampling, and just to note that she's moving into her second year of sampling now, and so lots more to come as well, but lots of exciting things to talk about for now. So go ahead, Taylor.
Hi everyone, I want to start off with a summary of my surveillance methods and dive into a lot of the interesting results that I obtained from my 2021 field season. So with over 20 years since the last survey, I wanted to make sure my methods were comprehensive in collecting a diverse range of species across the province, and therefore I focused on two types of traditional surveillance. So this included larvae collection as well as adult collection. So I had a few considerations when selecting sites to collect specimens from and to have a maximum variation between sites across Nova Scotia. So first I want to look at climate. As Laura mentioned, we have 9 ecoregions in the province, as you can see in the colored picture. It's defined in the province based into regions on temperature, precipitation, as well as elevation. I also want to look at proximity to human population. So I want to look at if I was finding mosquitoes in more remote or rural settings, as well as habitat type. So I want to look at the land classification of where I found these mosquitoes. So for example, if it was a wetland, permanent pond, or temporary pond. I also want to look at the seasonality of these species. So I want to see the time of the year that we see these species. Do we see them early in the season, later in the season, and do they have a defined temporal presence in certain regions of the province? You can go to the next slide, Laura. So during larval surveillance across the province, I used the dipper to scoop larvae at over 230 sites, including natural wetlands, artificial containers, and roadside ditches, and additionally recorded when larvae were not detected at a site. So in 2021, I sampled from June to October and sampled about every 3 weeks to coincide with the 2-week life cycle of mosquitoes. And during my sampling, I visited sites across the province, and my main goal was to observe habitat preferences for each larvae collected, as well as to determine if these preferences change throughout the season. So larvae collected at each location would be brought back to the lab in a Whirlpack bag, as you can see in the photo here. And these would be brought back on ice, and I would do a morphological identification of these species. So to double check my ID, I would also rear them to adults, as many larvae can be indistinguishable from each other as a larval stage. I can go on to the next slide. So to collect adults across the province, I used CDC light traps at 40 sites across the province from June to October. And you can see the sites represented by the red triangles on the map here. CDC light traps were installed with both a UV light to visually attract feeding mosquitoes, as well as dry ice. As when dry ice sublimes, it releases CO2, which is a universal attractant for feeding mosquitoes. So basically, it replicates the breathing of a potential meal, such as a bird or a mammal. A fan on these traps was powered by a battery, which sucked in any mosquitoes that were attracted to this trap, and I would be able to collect these. So these were installed usually overnight in the afternoon, around 4 to 6 p.m., and I would collect them again in the morning. And these times were selected to ensure we collected a high diversity of mosquitoes, including those that feed during the day, the evening, as well as during the night. I CDC trapped mosquitoes from July to October, and additionally, many traps were installed with what's called a Wattman card. So as you can see in the right side of this image here, a Wattman card would be baited with honey, and these cards are capable of collecting, deactivating, as well as preserving nucleic acids. So basically what this means is I allowed captured mosquitoes within the traps to feed on these cards. And this allowed me to identify viruses present within these trapped mosquitoes using real-time PCR. So for example, I could identify if any of the mosquitoes I caught were carriers of West Nile virus or Eastern equine encephalitis virus. I also additionally collected biting mosquitoes. So basically any mosquitoes that were attracted to me or my field assistant during the day, I collected those as well. So as I mentioned before, many mosquito larvae can be indistinguishable from each other. And therefore I would bring the larvae into the lab, not only to be identified, but to be reared to adulthood for a second ID. So distinguishing features in larvae include hairs in their head, which are called setae, which can vary in number and in placement within their head. Larvae also have a siphon on their tail, which is how they breathe air, similar to a snorkel. Many species have unique hair placements on the siphon, which is the feature that helps me when determining which genus the mosquito belongs to. For the adults, many species can be distinguished from each other based on the color and placement of scales across their bodies. So some species have alternating stripes of color or distinct black and white patches. Also, many Aedes species are distinguished from other genuses based on the striped pattern on their legs, with distinct black and white bands. On this slide, I have the larvae and adult stage of the species Culex Millinera, which is distinguished from other species by the numerous tufts of hair on their siphon, as I've circled there. The adults are a bit trickier to distinguish from other adult species, especially from Culex and Culecetta species, which supports the need for ID at both life stages. If samples are unable to be identified for any reason, for example, if they're damaged, I can send them away to be DNA barcoded, meaning that their DNA can be examined to determine what species it is based off a highly conserved region of the genome. I'm currently in the process of cataloging all of my species, creating a set of voucher specimens for each species I've identified to be confirmed by a trained entomologist, as well as a DNA catalog of each of these species using the species-specific barcode regions that I've mentioned. So during my collection, I collected over 4,000 specimens in 2021, and I actually detected 29 species, with 4 of these species being detected for the first time in Nova Scotia. Of the original species detected during the 2001 survey, all but 1 were detected. So the single species Anopheles walkeri was not detected during my 2021 survey, and this may be due to the delay of my sampling until June. My proposed start date of May was actually delayed due to a COVID travel restriction ongoing in the province, which may mean that I either just missed a species because it is an early season spring species, but this year I have already commenced sampling. So hopefully I will be able to kind of compensate that data this year to actually determine if it's here or not. So I detected 4 new species for Nova Scotia, and the first of the 4 species is Culasera Minnesota, which is originally from Eastern United States. It is not a known vector of disease, and it was found in Halifax, Yarmouth, and Annapolis Valley in June and September, and I found it particularly in dikes and roadside ditches. The second newly recorded species is Toxorhynchites rutilis, which is a tropical species rarely detected in Canada. So I found 1 singular larvae in the province in Cape Breton in July. And these species are unique as the adult females do not take blood meals, meaning the species does not vector any disease to humans. The third species I detected was Culex Salinarius, which is originally from the Eastern United States. And I found that in Halifax and Yarmouth in September and October. And I found this species particularly in containers and temporary pools late in the summer. This species is a competitive vector of Westernella and triple E as well. And finally, I detected Culeocetta Millinera, which is a species originally from the Eastern United States that is a very strong vector of triple E and Westernella virus. This species was detected in Annapolis Valley, Truro, and Halifax in bog habitats, and is a species of extreme medical importance, established as the most important bridge species of triple E between birds and humans in the United States. So in 2007, Aedes japonicus, which is a vector of Westernella virus and equine encephalitis virus, originally from central Japan, was first detected in 3 counties in the province, specifically Annapolis Valley, Truro, and Halifax in containers. So in 2021, I detected this species in all 17 counties, particularly as larvae in tires. Adults were detected at low numbers from June to October, and larvae were detected during the same period with a noticeable increase in numbers later in the season. This summer, I interestingly collected Aedes japonicus larvae as early as May 7th, which is noticeably early compared to many of the species I've collected so far in my study. This shows that since first detection, this invasive has quickly expanded in range across the province, and as many medically important vectors like Aedes aegypti and Aedes albopictus are also container breeders, this means that they may be able to show similar results if their range expands into Nova Scotia as well in the future. The seasonal presence of adults for the genuses of mosquitoes that I collected showed defined trends of absence and presence. Aedes mosquitoes tended to be present in my traps throughout the season and consistently were the species I detected early in the season, whereas species from the genuses Coulocetta and Wyomia were more restricted in their seasonal presence, with a clearly defined peaks in July and August respectively. Coulocetta perturbans is a voracious human biter during the day and a competent vector of Westernella virus and equine encephalitis virus, whereas Wyomia smithia is not a big human feeder, is not known to transmit any disease to humans. Understanding the seasonality, again, allows us to track how phenological shifts and understand seasonal shifts in disease risk across the province. So during the 2001 survey of the province, adult mosquitoes were not generally collected in high numbers until July, whereas during my sampling in 2021, I found a large number of adult mosquitoes from early June on until mid-October, although many of these discrepancies may be due to differences in sampling intensity. Additionally, the 2001 surveillance of the province concluded that vector species such as Aedes cantator were not found at high densities near residential areas, and that Aedes cantator and Aedes sollicitans mosquitoes were by far the most collected species in the province. During my sampling in 2021, I noticed that Culex perturbans and Aedes japonicus were the most abundant species, and both species are competent vector species. As Aedes japonicus larvae were associated most with containers, I found that density for the species was highest in residential areas compared to remote areas, showing that there was a clear link between potential West Nile virus risk and human populations. And this link was not really determined in 2001 quite yet. So during my surveillance, I identified some hotspots of disease risk in the province. So for West Nile virus, Kings County, Lunenburg County, and Halifax County showed the highest number of vectors for this disease in the province, with Kings County having the most of 12 species during the season. Cape Breton and Victoria County had the fewest vectors with 3 known West Nile vector species. For Eastern encephalitis virus, the risk was more uniform across the province, with still the lowest numbers of vectors in Cape Breton, Inverness, Victoria, and Richmond counties. Noticeably, Kings County had by far the highest number of EE vectors at 12, with Halifax coming in second at 10. This may be due to a higher number of human population, which could result in not only more food sources, but as Laura mentioned, a higher diversity of habitats for vectors to proliferate, potentially at higher numbers, and may also be due to the lack of intense sampling on my part in various regions of the province. This season, I hope to strengthen my sampling in areas of lower species detection to make sure I'm not just missing the collection of vector species within these areas, particularly in Cape Breton. So while sampling, I noticed clear distinctions between species by which habitats larvae would be detected in. So here in this graph, you can see that Aedes cantator seems to be a pretty opportunistic species, with larvae detected in all 5 habitat classifications. So in my research, I defined habitat as either a container, an ephemeral pond, which is a temporary pond, a permanent pond, a roadside ditch, or a wetland. Alternatively, Aedes japonicus shows a clear preference for containers in this graph, and in particular tires, with similar results in previous studies of the species. Culex pipiens larvae were mostly detected in ephemeral ponds and containers, and Anopheles punctipennus showed clear preference for permanent ponds and roadside ditches. This information helps us to make recommendations to the public and to understand where to manage species if that becomes necessary. With a distinct definition in breeding habitat for many species in Nova Scotia, I also wanted to look at if there were certain habitats that were more supportive of vector species. So in this graph, you can see there that although containers had the lowest diversity of larvae species, all the species detected were known vectors of West Nile virus or EE. So ephemeral ponds and wetlands show the highest diversity of species, and both had a large proportion of vector species as well. Container and roadside ditch habitats are a direct result of human impacts on the environment, and this may show that there's a link between the altering of natural habitats and vector species prevalence. So to move on to the final and the most ongoing aspect of my project, I want to look at eDNA. So this is a new novel way to detect invasive mosquito species, and it's called environmental DNA. Organisms leave behind genetic material or DNA in the environment through skin particles and waste materials such as feces and urine. For mosquitoes, we can determine the presence of larvae as well as identify which species are present through the collection of water samples from potential mosquito habitats, without having to collect the actual juvenile mosquito. Using a water sample, the DNA can be extracted, and using next generation sequencing, we can identify all species within that water sample simultaneously, allowing us to create a profile of the mosquitoes within that body of water. The method of collecting water samples to detect invasive mosquito species has been used to detect malaria vectors in Europe, and is shown to be quite accurate and comparable to traditional sampling. In 2021, I collected water samples from 34 of my larva habitats while sampling. About 45 milliliters of water was collected from each site using sterile pipettes, and I transported that back to the lab on ice. When larvae are present, I'm able to use eDNA to determine which species it is. And if there are no larvae detected using my traditional sampling methods, eDNA may be able to reveal which species I missed during traditional sampling. So I believe eDNA has the potential to be more time efficient and cost effective than traditional sampling, as water samples can be remotely collected. So this allows us to encourage community involvement in procuring water samples, particularly in remote and underserved areas. And they allow for a more comprehensive surveillance program in Nova Scotia. This season, I aim to obtain water samples through engaging in public outreach across Nova Scotia into more communities. And I think Laura will talk about that a bit as well.
Unmute myself first. Okay, thanks so much, Taylor. So, Taylor has collected really a wealth of data so far in the first year, and now we're going to supplement that with a second year and this eDNA work that she spoke to as well. And so from everything that she's just talked about, all of the species that we've been finding and the different connections we can make between which species are where and which habitats, this is leading us to a series of questions and hypotheses and other roads of investigation that allow us to better understand what the disease risks are going to be and what we might need to do to help alleviate some of that. So the first thing that I think about is, okay, so the number of species in the province is changing. We are seeing shifts every 20 years when we do our surveillance project. We are seeing a few more species crop up. But the interesting thing about some of the species that we see crop up is the differences in sort of how they're spreading across the province or where we see them and how often. So for instance, with Japonicus, we saw this really dramatic map that Taylor showed between sort of its first detection in sort of the mid-2000s to now, when it's really just, it's everywhere, and it's one of the most abundant species that she collects. And we sort of know this about Japonicus and its global invasion is that it's very good at invading. It does it very quickly and very effectively. But compare that to something like Millinera, which she also spoke to in this important bridge vector of different arboviruses. And it's the first time we found it in Nova Scotia. And so at first we were like, wow, that's really interesting. I wonder if it's expanding its range up here. But it turns out really that Millinera was recorded back in New Brunswick in that study that was done in the early 2000s. It was even found in Newfoundland and Labrador back in the 60s or the 70s, of one of those approximate decades. And so it brings us to a point with certain species of mosquitoes, ones that we're not necessarily seeing sort of take over the province. Are we seeing potential reintroduction of species over and over again? One potential, of course, is that sampling has just not caught these and they've been here for a while. But it's intriguing that we're only seeing Millinera here and now, and it's definitely one of the main vectors of EEE, and EEE only showed up in Nova Scotia within the last decade or so. So I would be interested to know if we're seeing repeated introductions of certain species that maybe are still just pushing their northward boundaries and haven't yet been able to completely establish, but might be able to stick around long enough until there's a really harsh winter, and then they sort of get knocked down and then they get reintroduced again. And things like that may happen with a lot of our human transportation that can carry mosquito eggs and introduce them to new areas. And I think of Millinera in particular because they overwinter as larvae. And like I mentioned, I'm always thinking about winter, and overwintering as a larvae is kind of a hard thing to do in a harsher winter scenario. And so that's one of the reasons I wonder about how we're seeing different species enter into our provinces. And so what that means for the future is if we haven't seen them very much, if we're just sort of seeing these little peaks of them from time to time in different provinces, have they actually established there? Well, we need our regular monitoring to tell us whether or not they're going to be here for good. Okay. Now, the other thing that Taylor talked a lot about here are, oops, if I can get myself to move forward, there we go, are these region-specific risks as well. And so we saw on some of Taylor's maps, again, these areas through Nova Scotia where we see really sort of heavy numbers of mosquitoes, high mosquito diversity, excuse me, in these areas and related to vectors for sure of human disease. And so some of the concern here being that some of those areas that she mentioned, so For instance, oh, I did find my laser pointer. So very excited. Here we go. We've got Kings County in this area and the HRM, excuse me, and then up through this area of the province where we've got sort of Truro. In these areas here where we're seeing these high diversity of species, a lot of vectors of potential arboviruses, and these are heavily populated areas of Nova Scotia as well. So the first thing this tells us is that we're seeing a lot of these vectors. This means that there's a lot of humans around. It means higher potential for contact with humans and potential disease transmission. We might think of this especially in any areas where if we're looking at transmission between birds and humans, especially the Annapolis Valley, being an area where we see a lot of bird migratory activity that could become a concern. And then the other thing to think about here is this, does this help us with any kind of control? So as Taylor mentioned, a lot of our container breeding species, here we go back to my dad's yard here where I go and I dump out all of his containers every time I visit, after I collect the mosquito larvae, of course, is this idea that we can really push, managing mosquito risk in some of these urban areas where, or across the province, anytime you have or anytime you're providing mosquitoes with a nice little place to lay their eggs, we can really tell people, make sure you're dumping out the containers in your yard, because these are the spots where the mosquitoes like to breed. So regularly go around any little spots, get those containers dumped out. And that's going to help tamper, hopefully, or dampen the number of these disease vectors that we're seeing in some of these heavily populated areas. The other interesting thing about this that Taylor made mention of that I wanted to talk about is this idea of human disturbance and its impacts on the kinds of mosquitoes that we're seeing and the abundance of these mosquitoes as well, and what this means for disease across the province and across the globe as well. So this is a recent and really interesting study that actually came out of Kruger National Park, so not in Canada, of course, but so this is out of Kruger National Park, and this study was able to show that in areas where there is a lot of human activity and a lot of human disturbance, so you might relate this, so for instance, to areas in Nova Scotia or across Canada where we're seeing, again, a lot of urbanization, a lot of development, a lot of disturbance of different ecosystems. What they were able to find is that the shift in the community of mosquitoes started to select for mosquito species that were more likely to be human biters and more likely to be vectors of disease. And as you can see in this graph here of mosquito abundance, so if you move towards the yellow, this is the disturbed areas and the blue is in the undisturbed areas. You see a difference in the community of mosquitoes, more vectors of disease and in higher abundance. And so we're going to work now to try and figure out if some of the patterns we're seeing in these populated areas in Nova Scotia have potentially anything to do with some of this disturbance as well. And this sort of brings me to this idea too of how we balance our conservation goals with our public health goals as well. So this was a question that was actually brought to me just a couple of days ago. Somebody asked me, they said, Laura, does no mow May mean more mosquitoes? So if we aren't mowing our lawns, for instance, and we have nice tall grass, does that impact mosquito populations? And I had to think, well, potentially, because it gives them a nice grass is this nice humid protected little area for them to rest in. And maybe some of you have walked through a tall grassy area in the summer and had a big cloud of mosquitoes come out at you. So they do find these areas. So potentially we think about this for ticks as well, of course, who also like nice tall grass. And so sometimes we think about this idea of conservation and public health maybe having slightly different goals, not goals, but slightly different mechanisms to arrive at their goals. And maybe these don't always align necessarily. But I think that last slide does help speak to that in some way that, again, if we think about this under our One Health perspective, if anybody's familiar with this, I'm sure anybody working in public health who's attending right now has probably seen this many a time, but this idea that we are sort of working towards understanding that this is our ecosystems and our own health are all need to be balanced within each other. And this includes things like mosquitoes and arthropod-borne diseases as well. So if we can do things like help to mitigate what's happening in terms of environmental disturbance, Well, maybe that does things like, maybe it helps to preserve the animals that are going to predate on mosquitoes and eat them up, and you know, help to make sure that we're not then also just selecting for mosquito species that survive really well in disturbed areas, who apparently also happen to be the ones that like to bite us and transmit disease as well. So I think interesting to think about, and a lot of collaborative work to be done here between disparate fields coming together to help us understand these connections and help mitigate some of the disease landscape shifts that we're going to be seeing. And I just wanted to bring up very at the end here, so this is again Taylor's eDNA work that's coming up here. We're working towards using this environmental DNA as a potential way of surveying for these mosquitoes throughout the province and assessing this disease risk. And so if you're in Nova Scotia right now and you're tuning in Mi'kma'ki, please get in touch with us. We're sending out some information now about how you can participate and collect water in the province and send it into us for some DNA sequencing, so we can work on this community science aspect of the project to determine if this could be a viable way of moving forward with more regular surveillance. So I just want to say thank you so much to all of you, first of all, for tuning in. Just a pleasure to speak to what I presume are human beings on the other end of the screen. But in any case, many thanks to you, and as well to our wonderful collaborators, many students whom have been essential to helping out with the field work, as well as Jeff Ogden with the province here in Nova Scotia, who was essential, the person who did the survey here about 20 years ago, and also helped us sort of get off the ground last year when some of our supplies were stymied by supply chain issues and COVID, as well as all of the different national and provincial and municipal parks and such around the province that have been really essential to us, and a bunch of different organizations as well, like Hypotheses Nova Scotia, the Mersey Tobiatic Research Institute, the Harrison Lewis Center, and then naturalist societies who were recruiting as well to help us with this community science aspect of the project. Okay, so I'm gonna take it to there. And yeah, Taylor and I would be absolutely delighted to answer any questions that you might have.
That's great. Thank you both for your presentation. Please, everyone, use the Q&A. You can use the chat if you want to ask questions. I'll get to those in a second. I just wanted to comment. I admire both of you for your bravery in collecting mosquitoes, because I think most of us would avoid it. And Taylor, in some of the photos where you're wearing shorts, I was thinking, I wonder if you no longer notice mosquitoes biting you. But maybe the bigger question is for both of you, a couple of things that I wanted to ask, and probably naive, but are there locations where you were collecting, you know, different, obviously, mosquitoes and larvae that really surprised you, that maybe you hadn't thought you would find many? I mean, one of the areas, obviously with the urbanization, and there's a lot more spots that I think happened, but are there any places that really surprised you? You thought there'd be a higher count, lower count?
Taylor, you go ahead with that one. I saw your mic on mute.
Yes. Well, the most interesting thing in Nova Scotia is that there's actually a species of mosquito that lays their eggs in pitcher plants. So that was a very interesting thing to learn about. And actually if you sample a pitcher plant, there's hundreds of larvae in a single plant. I thought that was the most interesting thing. As far as sites go, when I was approaching this project, I pictured any body of water a mosquito would live in. And so I was sampling lakes, and there's no mosquitoes in lakes, brutally. So that was something that surprised me. But another thing was very shallow containers of water. Even early in the season, like Laura mentioned, that toilet graveyard, there'd be very shallow water, and you'd see so many mosquitoes. And I would always think like, oh, it's so shallow, especially with it's like that's not going to kind of last very long. But that was surprising me, that especially shallow water was where we found the most mosquitoes.
Yeah,
I mean,
sure.
one of the comments early on, Laura, and maybe is like, you know, you guys talked about tires a lot, but you know, so Rebecca mentioned early on that playgrounds often have tires, or people put those up in their own yards. Is there other recommendations of things you would maybe recommend people don't do from a personal point of view, or even within communities from a development point of view?
Right. Yeah. So I mean, just looking at that chat there. Yeah. So I see that. So yeah, they have to have drainage holes, and that's a great point, right? So if you're going to use things like tires for tire swings, or like all kinds of different playground activities, absolutely. So anytime we can reduce standing water is going to help us try and control those populations. Yeah.
So one of the questions in the chat is, do you have any comparison between morpho-based identification and eDNA-based identification techniques?
Right, yeah, so that's a great question, and that's definitely what we're moving towards right now. So we're in the process of sending samples off to be identified, and Taylor was able to, last summer, was able to take samples from areas where then she also identified any mosquitoes that were present in that area morphologically, and then we'll be able to compare between her findings and what we get back from the DNA sequencing as well.
So Taylor, I know you answered this in the Q&A, but somebody had said, amazing work and great presentation. Have you considered applying an occupancy model to assess your sampling? And they did have a follow-up. There are also occupancy models for eDNA, which could be extremely useful in determining your sampling. So can you talk a little bit about your response in terms of the occupancy model?
Yeah, so with so much data, I was really kind of window shopping for lots of different models to apply. And I didn't really know kind of what models to really kind of look into, because there's so many variables, so many different species I'm looking into. But I actually had a quick Google while Laura was talking about the occupancy model. And it looks like it's kind of like a user-friendly model that you can apply, especially with species surveillance. So I think it is something I will apply here. Let me just get this up quickly. So it's a model used to account for imperfect detection of organisms in surveys and determine the probability of the true presence or absence of a species at a site, which is exactly, I think, what I need to apply to my model, because although I do get to about like 300 sites in my seasons, it's actually, when you think about it, really not much of the province at all, especially with all the standing water. So a model like this is definitely something I should probably draw into my data, just to kind of broaden the reach of what my data points actually represent as far as habitat and the location goes.
Yeah, that's great. That would be lovely. We've been looking at a lot of different diversity indices and things like that about how to model the data, but thank you for that. I definitely appreciate that.
So Scott asked in the chat, so regarding the water sampling surveillance, is it simply a detection of presence / absence of certain species? Is there some ability to assess species numbers to allow for calculation of vector risk index?
Yeah, so yeah, it is just for presence absence currently, but Taylor, if you want to elaborate on that, please go for it.
Oh, sure. So it depends on the kind of application we want to have in our research. So for example, I was kind of in between, do I want to go species specific detection? So maybe narrow down to 6 species and say yes or no, or do I want to get a complete catalog of the species that could be present? So with the direction we're going with a full catalog of the species, you can't really compare the ratio or the amount of species. You can only just do if it's there or not. Whereas the alternate approach is species specific. I could use quantitative PCR to really kind of have a standard curve compared to a known concentration of DNA and actually determine how much of that species is there and compared to other species, which is also interesting in its own way to maybe see how communities of species are represented in that body of water. But for our research, we're just doing if it's positive or negative, especially when the survey is so broad and over so many habitats. I think we really just had to narrow down to if the species was there or not, and also cover more species rather than just a few for qPCR.
Yeah, we were concerned with making sure that we also, we wanted to capture that whole biodiversity as well, because one of our sort of mantras going through is sort of a, we don't know what we don't know. And so if we only look for certain species, we have the potential to miss other things that come in. So, yeah.
I know there's lots of questions coming in. I just wanted to ask, and I think it popped up in one of these questions, but is like public health in Nova Scotia using your data? Have they talked to you about what you're finding and implications at this point, or is it connections yet to be made?
Mostly connections yet to be. So we are in contact with public health in Nova Scotia. They're aware of the project and we'll be definitely sharing our data with them. Currently, I don't think they're using the data currently, but I think as we enter into this second season and we finish up with the this next season of data collection in the eDNA, then certainly we'll be at least passing it on. So we'll see.
Great. So Josh says, great presentation. So competing priorities between ecosystems and human public health is a real concern. Some health professionals see no problem with spraying or fogging for adult mosquitoes with malathion and other organophosphates. How do we balance the need to minimize human health risk and the conservation priorities?
Yeah, it's a great question, Josh, and I think about this a lot. I don't have a really pointed answer, I suppose. So I think part of what we're finding is, again, if we can start to help people understand that conservation can actually help with control to some degree. If we did see that, for instance, if we do think, for instance, that the disturbance is going to lead to greater numbers of human biters and things like that, if we can try and, again, it's sort of moving towards this One Health perspective a little bit more, right? If we can help balance our ecosystem health, that should help to balance, balance the vector situation a little bit as well, or at least we would think so. We think about this with ticks as well. For instance, you think a lot of we see links between certain invasive plant species and tick abundance, and things like that. So, preserving our habitats also does things like it increases the number of different vertebrate species that are present there. And so that also helps sort of, the mosquito species that tend to be the bridges between humans and animals. Well, if there's a lot of other vertebrate species around, so, other wild mammals and things, then maybe we won't see actually quite as much of them jumping over to us. If they've got enough hosts in the wild, in these protected ecosystems, and there's more sort of dilution of the parasites throughout all of these vertebrate hosts, then that can sort of help reduce our risks as well. And then there may be areas where, that's the thing, right? I also come to think where we're still pretty protected here. We don't see a lot of mosquito deaths here versus other areas of the world. And so there may be some pockets of time where more severe control measures may be necessary to prevent human suffering. But I think it's really going to be about a balance of trying to marry the two, conservation and public health, together and see how they can actually augment each other rather than be fighting.
So I know we're at our time limit, but I wanted to just see if you guys were okay to keep answering a few more questions, and we'll keep the recording going. Okay, great. So I'm going to jump over to any surprise detecting C. Millenera vector for ectoline encephalitis in Nova Scotia. Any public health data to link to the presence of this location,
Yeah, so I was speaking, so, it's definitely, we said with triple E, and triple E only showed up in Nova Scotia within the last 10 or 12 years, I believe. And so I guess I'm not surprised to see it in some ways, because we've had triple E show up here and it being the predominant vector. In some ways I sort of went, oh, I guess that makes sense, right? Maybe it has been here, and maybe it's linked to, maybe its introduction into Nova Scotia or its establishment here is linked to also that sort of that rise in triple E in Nova Scotia, or the introduction of triple E to Nova Scotia. So that's a possibility. And also the fact that it's been around, it was found we hadn't found it in Nova Scotia before, but it was in New Brunswick, 20 years ago. We had that odd one in Newfoundland and Labrador a few decades ago. So yeah, not totally surprising, I suppose. I'm expecting it to be okay. Yeah, that it seems very reasonable for it to be here, especially with that link to triple E showing up in Nova Scotia.
Okay. So Michelle asks, were there relationships between sampling sites, example, GPS location, landscape, and species diversity?
Did you want to take that? Is that, sorry, I'm reading the question too here. Sampling sites and species diversity. Yeah, so certainly I'll probably let Taylor take this one. She'll be a little bit more familiar with her specific sampling sites and those diversity. But in terms of the GPS location, I'm not sure that we've gone as far down. We haven't drilled quite that far into the data yet. And should we go beyond some of the sort of county areas? But definitely we're seeing some of those relationships between some of these larger geographic areas. But Taylor, go ahead if you've got some more insight into the more specific GPS locations.
Oh, I don't really have anything on the specific GPS location, just because a lot of the times my sample sites would be kind of within proximity to each other. So I could kind of correlate there. But I did find that in northern Amherst, particularly around the provincial park there, that there was quite a bit of diversity in July. I did notice in one of my CDC traps that I set up, I got 15 species in one day at one trap. So you could say that the diversity was particularly high at that GPS point. And it does make sense as this provincial park, it's right on the sea. So you have a lot of, you have the saltwater species such as Aedes cantator and Aedes solicitans. And then you also have, you know, like Laura was mentioning, you have human populations. So you have a wide diversity of habitats. So there's definitely artificial containers for like ditches and wetlands as well. So I just think for that GPS point, for that trap, there was a very high diversity. It might have also been a correlation with the perfect temperature for all the diversity of the adult collection. But when it comes to larvae, if this is what the question also is referring to, I did find that larvae sampling, the diversity at each site was lower compared to the diversity of adults that I would be collecting. But this could also be just due to the fact that these traps have a pretty wide radius of collection or attraction to the mosquitoes, whereas my traditional larvae sampling, it's just maybe a few meters of collection compared to hundreds of meters for one of these CDC traps, for a single GPS point at least.
And I just wanted to add that that really speaks to sort of this idea of where we are in the Maritimes as well, being that we have a lot of sort of our towns or city centers located coastally as well. So we're a nice warmish, humid area with all of these lovely salt marsh and you know, flooded areas that are close also to our heavily populated areas that also contain our containers and those kinds of things. So we're an area of Canada, especially that just provides a lot of good mosquito habitat that humans also like to inhabit those areas as well. So we sort of are at the intersection of a bunch of different kinds of mosquito habitats.
So I'm going to paraphrase maybe two questions here in my little add-on. So Sydney was asking about, you know, provincial data around the West Nile virus that might add to the evidence base. And I had the question at the same time when you're showing the map of the expansion from, you know, a few counties to the entire province. I mean, has there been data linking, you know, EEV and West Nile virus to actually what you're seeing with the populations, or is that work yet to be done?
It's a good question. So I think that there's definitely been West Nile virus surveillance on and off through Nova Scotia. The cases, at least in humans, have been fairly low over the last several years now. We sort of had peaks, I think it was like 2003, 2007. And then, you know, it hasn't been quite as predominant, but we're hoping that we're going to be able to maybe pick up some of this with our Whatman cards as well, or sequencing some of the mosquitoes so that we can see if we're actually detecting West Nile virus in any of, for instance, the Japonicus, or the, you know, look, are we seeing EEE in the Millinera in the traps that we catch them, and can we actually detect the virus as well? Because I think absolutely that's the point, right, is we want to be able to make some links between seeing these vectors here and whether or not we're seeing the viruses as well. EEE is an interesting one because now that we know we have it in the province, I was talking to some veterinarians about it, and they were saying that, you know, they just vaccinate the horses through the province. So hopefully that helps with, you know, some of the sort of reservoir hosts in the province. But of course, it's coming from birds as well. And we predict our EEE from bird movement up from Florida. And so we kind of get a forecast of what it's going to be like in Nova Scotia based on what happens in Florida in the spring. But yeah, no, I think you're absolutely right. And so we're hoping that we'll be able to make some more of those links, especially with the virus data that we're going to be doing some sequencing with as well.
So maybe Michelle's question is a bit of so are there intentions, I mean, whether you know of or within your project, to for performing arbovirus testing, particularly beyond EEE and West Nile virus, such as orthobunya viruses?
Yeah, that's the thing, right? So I think like currently any virus testing has been pretty targeted as far as I know. If anybody knows that the public health's been doing things beyond West Nile or EEE, please feel free to correct me, but I think it's been fairly targeted. With ours, we're hoping to be a little more wide-ranging, not looking for just the specific ones. I'd like to send our samples off for next generation sequencing and be able to just characterize what viruses are there. But whether we'll be able to detect them all, I'm not sure. But certainly I think that's really important because again, it's one of those we don't know what we don't know. So definitely good to include.
So final two questions. First one from Lydia. Were you able to correlate your surveillance results with precipitation events, example, after extended periods of rain? Well, we expect seasonality, cold is not mosquito friendly. What might be a temperature threshold that enables certain species to thrive?
Mm-hmm. So yeah, so that's one of the things we're working on now, is trying to put some more of that climate data into marrying that sort of to our diversity and abundance data, and take a look at that, because certainly you would expect that you're going to see seasonal differences based on, you know, how harsh the winter was, and whether, what kind of precipitation we had, and do we see sort of dips in certain months, like early in the spring. If it's dry, that might delay the mosquito season, that sort of thing. And so we're going to be looking into those kinds of things for sure. Very important. And in terms of sort of things like temperature thresholds that might limit mosquito distributions, well, it's a little bit species specific, to be honest. And actually, there's some really interesting work that came out of York University, I think. Yeah, I think it's York, a couple of years ago now, actually showing the capacity for a species like Aedes aegypti to be quite plastic in its physiology in terms of low temperatures. And if you allow them to sort of acclimate, you allow them to get used to low temperatures for a little while, then you actually see their cold tolerance shift a little bit, and they actually become more cold tolerant and better able to survive low temperatures by a couple of degrees even. And that can be the difference between a mosquito being somewhere and not. So it will be a bit species dependent. They're all going to have their physiology is all a little bit different. You know, the temperature at which their bodies freeze might be a little bit different. The the temperature that they can withstand before they lose neuromuscular control is going to be different. The temperatures they can withstand before they die for reasons unrelated to freezing, things like that, varies between species. So I don't have sort of just a across the board temperature threshold for mosquitoes, but certainly there are thresholds for particular species, temperature thresholds that we can do. For instance, in the lab, we can work with those species and characterize their cold tolerance, and then make predictive models based on that. Yeah.
So one final big question for you. So how can the findings of your work impact city planning, both built and natural environments? It might be really hard to, sorry, that just moved on me. Okay, I'll read that again. How can the findings of this work impact city planning, both built and natural environments, for protection of planning as part of public health?
Oh, that's a good question. That's a big one too, right? So I think, again, this is going to be related to, I guess, to understanding mosquito habitat in general, and can we, how do we, gosh, I keep wanting to say, like, how do we minimize disturbance in our planning to, to help increase, Taylor and I obviously both have sort of a conservation spin on our ideologies. So that, it's a big question, I think, and I don't think I have the full answer to it yet, but I think that would be something really interesting to explore, especially with city planners, right? To be able to help them to understand, what disease risks might be associated with this. And so, obviously, if we can do any kind of planning, again, to reduce standing bodies of water that don't facilitate other kinds of biodiversity, like fish and frogs and other things that would eat mosquitoes and help keep them in check, and other things that we like to have around. But yeah, anything we can do to minimize those aspects. I guess part of it would be, part of it might be, don't build your towns next to salt marshes, but that's sort of out of the question here in Nova Scotia to some degree in a lot of our areas. So it's a big question, and I think it's a great one. But I don't have a full straightforward answer for that one. I think there's a lot of different moving parts there, but definitely something that we need to consider.
That's great. So I realize we are quite a bit over our time. I just wanted to say thank you, Laura. Thank you, Taylor, for a great presentation and answering all those questions and staying extra time. We really appreciate it. And thank you, everyone who attended, and we appreciate your questions. And with that, we'll say thank you and have a great day.
Thank you all so much, and feel free to send us an email if you have any other